In drug research, being able to reach the right target with the right therapeutic benefit is challenging. A team of researchers at the University of Queensland has found a strategy for enhancing the small, potent subunits of proteins, known as peptides, with sugar groups to make them more precise and responsive as therapeutics. They may be used to target respiratory, neurological, and metabolic disorders, among many other applications. In the recent study led by Istvan Toth, their strategy involves attaching sugar molecules to peptides, which can mediate cell-specific delivery by targeting sugar receptors inside the cells. This process of adding sugars to compounds is known as glycosylation. Glycosylation has been found to strongly improve a drug's ability to get inside of the cells and stay active in the body for longer. Toth's team wanted to see if glycosylation of the peptide enkephalin, an opioid used to regulate pain in the body, could help it bind to a sugar receptor found in the brain called ASGPR. ASGPR is an important target because it can mediate cell-specific delivery by binding with peptides and fats that have attached sugar groups. When more enkephalin binds to ASGPR, the opioid's effects become stronger. All you need to do is mark the letter with the correct postcode to ensure it makes it to the right address. Using a one-pot reaction, an efficient process whereby a substance can undergo successive chemical reactions in just one reactor, they mix the enzyme UDP-glucose, important in metabolism excretion, with the enzyme galactose epimerase, which converted the attached glucose on UDP to a galactose, making UDP-galactose. UDP-galactose then went on to react with the enzyme LGTC. Involved in transferring sugar groups to specific sites, LGTC swapped the galactose on UDP and attached it to enkephalin. All in all, they took two sugars known to target the receptor, galactose and lactose, and stuck them to enkephalin, making the following glycopeptides: Gal-Inc, Lac-Inc, and Gal-Lac-Inc. After synthesis, they tested their compounds' stability and permeability by mixing the glycopeptides with human plasma and colorectal adenocarcinoma cells, known as CACO2 cells, separately. In human plasma, the sugary peptides stayed effective for a longer period of time. They became more stable than the naked peptides. In CACO2 cells, the sugary peptides had similar results. Permeability increased as the peptides passed through the membrane layers of the CACO2 cells with ease. This increased stability could be due to the compound steric effects, whereby the added sugar groups occupy more space in the peptide, shifting its shape to a point where it becomes protected from enzymes that would otherwise degrade it. These compounds then went on to bind to ASGPR, which the team analyzed using molecular docking and SPR experiments to give snapshots of the binding interaction. Similar to assembling a jigsaw puzzle, the glycopeptides fit together with ASGPR in a very specific shape and orientation. Both tests confirmed that adding more sugars to enkephalin increased the binding strength of the peptide to the binding site ASGPR. While further experiments need to be done to confirm therapeutic potential, Toth's group of researchers thinks that this technique can be more widely used to enhance peptide drugs to contain different sugars for targeting other kinds of sugar receptors for patients to metabolize medicinally.